So today we will discussing with Dr. Elzbieta Iwaszkiewicz-Eggebrecht and Dr. Andreia Miraldo about the recent PLOS ONE Lab Protocol, FAVIS, fast and versatile protocol for non-destructive metabarcoding of bulk insect samples, and the associated protocol in protocols.io. Ella and Andrea, thank you so much for being with us. Ella studied biology and obtained her master's degree from the Sorbonne University in France. She followed it with a PhD in evolutionary biology completed at the Max Planck Institute in Germany. She is currently a postdoctoral researcher at the Natural History Museum in Stockholm, Sweden, where she uses DNA-based methods to monitor insect community changes. Andreia Miraldo is a research associate at the Swedish Natural History Museum in Stockholm. Since 2018, she has been acting as the research manager at the Insect Biome Atlas, a large-scale insect biodiversity research project, looking at characterizing with unprecedented detail the insect faunas of Sweden and Madagascar. Andrea has a PhD in evolutionary biology by the University of East Anglia in the UK, and before moving to Sweden, she held postdoctoral position at the University of Helsinki and at the Natural History Museum of Denmark. Thank you again so much for being here with us.
Thank you for having us.
To kick off, can you briefly explain what the protocol is about and how to use it and why it is relevant?
Yeah, so I'll start. Our protocol describes a non-destructive way of processing samples for a DNA-based metabarcoding of insects, mostly for biomonitoring purposes or biodiversity discovery. So the relevance of this is that basically insects are the most diverse and abundant group of animals on Earth. They are in fact so diverse that the traditional methods of looking at animals, identifying their morphological traits and deciding what species that is simply not fit. Currently, we have around 1 million insect species described, and many of those are very small, inconspicuous, brown, grayish, and is indistinguishable to an untrained eye. And identifying them demands a huge expertise, which is a really scarce commodity, and takes a lot of time. So, and to add to this, actually, we know that this 1 million insects, it's only 1/4 or 1/5 of the total diversity. So there's a lot more to still discover. And this is why basically the traditional methods don't perform very well. And more and more methods that were initially devised for microbial communities, they are being applied to insect communities as well. So basically, you can collect insects by some trap, for instance, Malaise trap, like we do, where you have a huge number of insects in one bottle with ethanol. And from those samples, in our protocol, we describe all the way from those samples to basically DNA sequences that you can decide based on those sequences what species are inside, and in this way describe those species communities in space and time. So it's a very powerful method that gives quick access to the information about the whole insect community.
And when you mentioned that the previous methods weren't totally adequate, is it because they weren't good at do bulk samples, or because they were destructive, or both?
Yeah, well, I can take that one. Yes, so these methods, DNA-based methods for biomonitoring and biodiversity projects have evolved quite recently and quite fast. And traditionally, in terms of when we're speaking about insect biodiversity, these methods have been mainly traditionally used this what we call this insect soup. So they were what we call destructive methods. So they could basically make a soup out of the sample that Ella just showed you, and then they extract the DNA from the bulk sample. And you also already have early on, you had also non-destructive methods but those methods were mainly involved. You had to sort your sample. So you will, with the sample that Ella showed you, you will divide the sample with the large insects, medium insects, and small insects. And of course, this takes a lot of time. And it means that basically, instead of one sample, one DNA extraction, for each sample, you will have to do 3 different extractions. So it's time, it's very timely, and it's also costly. And also, you know, when we devised this protocol, for us, it was very important that we would keep the insects intact. This is because, first of all, we work at a museum, and we know that we spend a lot of effort and time in collecting these very important samples. So it is important for us to keep them as intact as possible. But also that we create a protocol that is fast enough that we can analyze these samples in real time to make differences and to be useful if you are trying to monitor insect populations in real time. So yeah, basically the protocols that existed were not really fit for purpose.
I see. And so the innovation is bringing together the idea of not having to sort the samples, that you can treat them all together in the same, not soup, but, you know, intact, but in the same mixture, and without perturbing the physical properties of the sample.
Yeah, and also we really wanted to keep the insects always in the sampling bottle. So basically, with our protocol, the insects, they never leave that bottle. They are always there. We do the DNA extraction there. After we extract the DNA, we refill it with the ethanol, the same ethanol that they were collected in, and then they just stay, and they stay in the collections for long term and for other studies. This is really important when you are working with insect biodiversity, because most likely all the specimens, or a large proportion of the species that you are going to find, are either unique for the region that you are studying. So there are new records for that region, or they are most likely quite a lot of them will be unique to there will be new species to science. So it's really important that you are able to go back to the sample and retrieve those. If you say in the sample that Ella is now holding, you know that you have a new species of bee, you can go back to the original sample and you can retrieve all the bees. You send them to taxonomic experts, taxonomists, that will validate or not your findings. So it's really important for us to leave everything in the original bottle and as intact as we can.
I see. And I would like now to get a little bit more into the details of the protocol. So while you were developing the protocol, was there anything that you tried and didn't work, and therefore didn't make it to the final version of the protocol? And I'm asking this question because, you know, usually negative results are done over and over again because people don't report them. So to prevent that from happening with the users of the protocol, is there anything that has been tested that you know doesn't work?
I can, not specifically in terms of negative controls. Maybe Ella will, whilst I'm speaking, maybe she will think of something that is escaping my mind. I'm thinking more in, of course, when you are optimizing a protocol, there's lots of things you test. It's like creating a new recipe for a food recipe in the kitchen. You try things work, things don't taste good, so you try again, and it's a little bit like that. So we did try lots of different tools and methods because we are working with a sample that has a specific size, and we want to be able to leave the insects always inside the bottle. So there are lots of things that we had to troubleshoot along the way, and those results, those little optimization steps, they don't really end up in the final protocol. I'm thinking of the very first thing we had to decide was how are we going to decant the ethanol, get rid of the ethanol from the sample and just leave the insects in a way that is fast enough, but also that we don't lose insects along the way when we are getting rid of the ethanol. So we came up with this mesh, a nylon mesh that we attached to the bottle opening. And of course, when you enter the world of nylon meshes, you have all different sizes of pore sizes, from very large to very fine. So we had to test different pore sizes, and some of them we tested with the tightest ones, and of course it would take forever to decant ethanol. So in the end, we wouldn't have a high throughput protocol, but a very slow one. So we then ended up choosing one mesh that was a compromise between getting rid of the ethanol in a quick enough fashion, but also making sure that we wouldn't lose the tiny little creatures that are so important for our later analysis. But in terms of negative results, I'm not sure. Ella, do you have anything to add?
I think it's a problem like with any other publication that you just show what works and maybe making a note, like sort of explaining, like Andreia did now, that this mesh that we propose is a trade-off between this and that, but we don't say that we tested this, right? Because it's just a lot of information. But maybe something to keep in mind. Yes, it's important during development.
And then going, like changing a little bit the question, but what is, in your opinion, or are, in your opinion, the most critical steps when performing the protocol? If somebody wants to implement it from the scratch in their own lab, what will be your advice?
Andreia.
I can take. I think it is, because this is what we call the mild lysis protocol. It's really important to measure the biomass of your sample, so that you know exactly the amount of lysis buffer that you add to the biomass. And this is important because if you add too much lysis buffer, what will happen is that you will lose the capacity to detect the tiny little specimens, because they will leak very little DNA, and your DNA will be too diluted if you had too much lysis buffer. So it is really important to be very accurate when you are measuring the biomass of your sample and add the proportionate amount of lysis buffer. Other things that are important is in terms of the incubation time. This is a mild lysis protocol. We say that you should incubate your samples for this enzyme proteinase K to lyse and to be able to release the DNA into the lysis buffer. It's very important that you are working with samples that are very small and samples that are very large. So it's important that all of the samples are treated in the same way, so they are incubating exactly like that. And sometimes even worse. I'm now pointing at Ella's a very small sample and a very, very large sample. So it's important that they are incubating exactly the same amount of time to minimize biases then in the data that you will have. So I would say measuring the biomass is quite important, and adding the proportionate amount of lysis buffer to that that sample is very important.
Because in the video, in the recording, only shows the person who's speaking. We don't see everybody simultaneously. Sorry. Ella, can you speak and show us the two different sizes or the range of samples that can be handled?
So yes, I brought two examples of samples. So this would be, yes, some samples are big or much bigger than this one, actually. And some are this small, just few insects inside, or even smaller, of course, because we also sample in winter in Sweden. So some samples are really tiny. And if you would add the same amount of buffer to those two samples, this would be just very diluted DNA. And then you would not recover the biodiversity that you're aiming for. So it's crucial. So many, yes, crucial steps. I think maybe also adding to this, I could say that in the molecular laboratory work, it's also important that from each sample, we want to catch all the DNA representing different species. So we tried, and some protocols, some researchers advise doing triplicate, so 3 different PCR reactions per each sample, just to be sure that you're catching all the DNA. And this is a lot of work. Also costs, but mostly, yes, it's a lot of work. So we thought we'll just add more DNA to PCR. But that didn't work because the buffers that you incubate them in are full of inhibitors. So just the reaction was blocked completely. And for that, we made the decision to make a sort of a trade-off where we increase the total volume of the PCR reaction, which is quite costly, but then we were able to basically add those few microliters of DNA that were necessary, and do only one PCR for this one sample. So I think it was a nice trade-off that was also a result of many tests, trial and error.
And the protocol and the article were published less than 6 months ago, but I want to know if you have tried to tweak it or improve the protocol any further since the time of publication. Have any changes been implemented to the protocol?
Not really, no. I think we've published it at the also point when this protocol has been used, tested, polished, and we were really, this was our final version. So it hasn't been tweaked, but I think that's a great feature of protocols.io that you can add onto it. And I think with next projects, I aim to publish earlier, put the protocol out earlier, and get feedback from the community, and then just publish next versions.
Yeah, that's a great point. I wanted to add that when Ella says that we have, when we published the protocol, it was quite well tested. In fact, we had processed already more than 7,000 such samples. So it was so well oiled and so well tested that we had already produced this huge data set. And we continue. We are now in more than 10,000 samples that we have analyzed using the protocol. So it really works well, and it has really been employed to thousands of samples. So we are quite happy with it. But I agree with Ella, perhaps having published it earlier on, we will have solved quite a lot of troubleshooting by having the input of the community.
Now, since you are asking about input, I would like to ask 2 questions. So the first one is about the motivations, not only to share the protocol in the platform, but have it peer review. And second, about what was the impact of the peer review process? What was the feedback that you received, and how that shaped the final version of the protocol? I have to say that the protocol looks beautiful. It's very didactic, it's very, very carefully crafted and very instructive. But go back to the question. Let's go back to the questions.
Okay, so maybe I will start. Well, thank you. I think the motivation to publish this protocol and publish a paper accompanying it was that, well, of course, sharing knowledge is a goal of every researcher. And that's obvious. However, those lab protocols tend to be often overshadowed by big results and big research papers. And in every life of every young researcher, they tend to get low priority because there's this next big result that you're chasing after. And I think. But I think it's important. I think the PLOS ONE paper basically gives a nice additional motivation to put it all together, to make it really nice, to make it accessible to people from the outside, and also just to collect information from everybody involved in developing of this protocol, because there were many people and we have quite few authors. And I was also very happy to have the ability to give them the credit they deserved, because this is often work, hard work that's not being recognized. So I thought that was a great way of having this. And then of course, publishing PLOS ONE paper gives more visibility and credibility to this method. It really widens the impact.
And can you comment on the feedback that you might or might not have received from the reviewers, yes, and how that helped shaping the final version, if any?
Yes, sure. That every peer review process helps polish and like elevates a bit the protocol and the paper. So it really definitely helped make it more accessible, more logical, and like general structure. Oop. However, I think we did a lot of work beforehand, because peer reviewers, the reviewers were quite positive.
And then the final question on my side would be, talking to both of you who are like early career scientists somehow, what has been the impact of having this protocol and associated paper published for your careers?
Yeah, I think as you mentioned, it's perhaps a bit early to know the impact. The protocol, I think, was published online in July. So
that's correct.
4 or 5 months ago. So it's perhaps a bit early, but we know for sure that we are having a positive impact in the research field, because we have been contacted by other researchers that are now using our protocol. A big research initiative, the BioScan Europe, which is a large European consortium using DNA methods for biomonitoring, is using our protocol. So we know we are having a positive impact, and this is exactly what we wanted. We wanted a protocol that was standardized and to encourage the community to use our protocol, because this, of course, is very important in the future. If you use always the same protocol, everyone is creating data sets that then we can combine and analyze together at a later stage. So this was really important for us. But yeah, let's see how in 1 year time where we will be. I don't know for Ella. I haven't even checked if we have had already citations or not. But it definitely gave us visibility, and we are being contacted by other researchers and other large-scale projects. So that's quite positive.
I also haven't checked, but I agree. Yes, I do get more messages with direct questions, and it's even a resource for me. I mean, I refer to this online version of the protocol all the time.
Well, you're right. It's early to say, but I'm happy that you have steered something in the community, and that you're being contacted. So before we wrap up, is there anything that you would like to mention that we haven't touched?
I think we're fine. I'm fine.
Not really. I think it was a very insightful set of questions.
Thank you. So with that, we will say farewell, and thank you so much.
Thank you.
Thank you, Gabriel. It was nice talking to you.